And the next speaker is Dr. Fang-Chi. And Dr. Fang-Chi obtained his bachelor degree in Taipei Medical University about the public health, and he went to U.S. New York University and obtained the MS degree in the bioengineering, and then he went back to Taiwan and to get the PhD degree in the translational medicine of Academia Sinica Taipei Medical University. And now he has been a research fellow in several university institute, including the Harvard Medical School and also in National Yangming Jiao Tong University. And now he's a research fellow of the Sino-American Cancer Foundation. And today he will give us a very interesting topic about the mechanistic insight into the GBE1, about the metabolic reprogramming in colorectal cancer. Let's welcome Dr. Xu.
Good afternoon. Thank you so much for Dr. Yang's kind introduction. My name is Fang-Chi Su, and it's my great pleasure to have this opportunity to share our current work about the GBE1-driven metabolic reprogramming in colorectal cancer, which is a very interesting part. We will see a little bit hint of the switch point of the metabolic reprogramming. Also, I hope in the end of this talk, I can try to show you several data-driven pipelines about our infrastructure. Okay. So before we start to talk about the work, allow me to introduce myself and my team. So I'm currently a scientist in bioinformatics and computational biology. Along my training path, I spent several years in university and institutes, both in the United States and Taiwan. So our team is focusing on the data-driven drug discovery. We use the large-scale population-based data, trying to find out some hint and go through the detail, integrate the structural biology, trying to deliver our digital discovery for the real life. So back to some backgrounds. Why colorectal cancer so matter? So everybody say that colorectal cancer is so severe. That's like a third place or fourth place, the lethal cancer type. But the growth of the incidence is very huge. The WHO cancer incidence projections just published this data. They say they project about 70% new case growth worldwide by 2045. Not only this, we are also this data also catch our eyes that Asian population got the huge burden. That's not only like a 70 to 80 growth in the Asian population, but also there's almost the most dominant populations in different countries. So Asia is very important. Not only this, when you see the mortality even worse. That's the Asian population got the worst mortality in the colorectal cancer. Surprisingly, not only the conventional colorectal cancer, but also early onset colorectal cancer has the similar pattern. So in the future of the world, we are thinking about Asian population is very important. We are trying to solve this problem, which is threat our life. So let's give us a hint. Like, okay, we know the progression. We know that's very important. Why that's so important? So in several published data, they said, okay, metabolic and the diet probably one of the reasons why Asian population gets so severe colorectal cancer. Not only this, but also the metabolic patterns keep changing during the tumor genicity. So from the from the normal to the pre-malignancy to the early stage to the late stage to the finally to the invasion and the metastasis. They have the colorectal cancer tumor cells obtain the different metabolic status. But also we found a key. That's when it's getting the worst prognosis. We found that that's and out of the glycogen and the carbohydrate metabolic pattern, the signal getting down, reach the peak, then switch to the fatty acid and the lipogenesis. So this one get us a hint. Okay, we know in our populations, metabolic is very important. That cause the high risk of the colorectal cancer. Not only this, but also once we can use this pattern to find the weak spot, everybody can think about how to kill a snake. You are killing the neck of a snake. So we are trying to see where's the weak spot and moderate this signature using a feature to leverage and generate a new therapeutic guidance for the moderation and maintain the good treatment outcomes. So we back to the population-based studies. This is our originate. Our idea originated from this study. It's the large-scale European and East Asian ancestry multi-omic GWAS study, but not only GWAS, but also PWAS. We can find not the conventional driven mutations driven variants. Also, there are six different genes indicate the glycosylations and the lipid metabolism. Afterward, in 2025, we found the following studies used the transcriptomic analysis and the EQTL-based Mendelian randomization. Upon these six risk genes, only one gene get caught the causal inference, which that one is GBE1 genes. So elevated of GBE1 genes and the variant of GBE1 genes can both increase the risk of colorectal cancer. But why is so important? Because these are all genes only enriched in Asian population. So this evoke our curiosity. Why? So we love starch. We are Asian population really love starch, rice, noodles, but not only this. So that's definitely something happens in the glycogen metabolism. So GBE1 itself is actually the glucogen branching enzyme. So which one is using the catalytic domain and the two carbohydrate binding domain, trying to elongate the glycogen from the oligosaccharide to polysaccharide, not a linear form, but the branching form. So the function, the enzyme function look like this. Using the carbohydrate binding domain to cut a oligosaccharide, then adding to the long chains in the catalytic domain. So when we are going to target this gene, the catalytic domain is the key to inhibit the function. Not only the metabolic function, but also in several studies has already reported the value of GBE1, especially in the non-small cell lung cancer, pancreatic cancer, and the glioma. So in this data, non-small cell lung cancer shows the very promising one when you are going to knock down the GBE1 expression. They have the lower energy levels, energy consumptions, and also the lower tumor progression. So it seems like one of the possible targets, but not really reported in the colorectal cancer. Is that a selection bias or just like a hidden genes we haven't found it? So that's our data. From the TCGA, you will see a very limited borderline differences between the tumor and the adjacent normal. So it's just a little bit elevated. And if you check the TCGA large scale data set, GBE1 didn't have reached the significance of the survivals. But in the several individual cohorts, you definitely see the worst prognosis in the high GBE1 expression patients. Also, our histology also provides our evidence to support that. Okay, we definitely see there's a high expression, high intensity of a GBE1 expression in tumor part. So we use a quick model to address that. Before that, in the previous biochemistry research, we already has reported that GBE1 is a downstream of a HIF1 alpha. So hypoxia triggered the GBE1 level. So in the acute hypoxia, we see an elevated of the GBE1 expression. If in a knockout cell, there's no different change. So that's how the really high correlated between GBE1 and the HIF1 alpha. So this is like one of the response of after the acute hypoxia. So using the hypoxia transcriptomic data shows, we see there's an elevated of the glycosylation, glycolysis, and the carbohydrate metabolic gene upregulated in the acute hypoxia. And there's a downregulate of the lipid metabolism. So let's indicate one thing. Okay, so acute hypoxia is seems like in the early switch part, right? From the tumor genetic landscape, maybe from the stage 2 or the early stage to the late stage. So it's consist with the previous data of metabolic analysis. So here we make a summary of the canonical GBE1 function. GBE1 was controlled and regulated by the HIF1 alpha pathway. So the production of the GBE1 can inhibit the FBP1, which is the intermediate genes for the fructose phosphorylations. The downregulate of this can activate the NF-kappa B, also activate the glycolysis to reach the tumor survival. So this is a hypoxia canonical pathway. Surprisingly, we're trying to see, okay, when we are reproduce the knockdown experiments, what happens? So we use the CRISPR-Cas9 model to knock out the GBE1 in several different colorectal cancer cell lines. We found that in several particular cell lines, they have a really good inhibition of the tumor proliferation. Also, the real-time PCR and the web bench experiments shows, okay, we see there's in the regulatory effect of the Wnt signaling. So why going down of the GBE1 expression can affect the Wnt signaling and reach to the inhibition of colorectal cancer proliferation is one of the questions we are trying to address. This also, we found another very interesting thing that our chip sequence and ALTA-seq data shows there's not only HIF1 alpha directly binding to the hotspot of the chromatin accessibility. We surprisingly found the PPA gamma and the RXR beta also got the direct binding in the promoter regions of the GBE1, and the RNA-seq data also shows there's a high correlation of this. So we also make a deduce, like there maybe have a secondary driven signaling from for the PPAR gamma. And we have some data that shows the correlations. Also, we also found in the IHC, in the stage 2 and stage 3, they have the unexpected elevations of a PPAR gamma. So PPAR gamma is one of the modulator and the regulatory signaling for the lipid metabolic. So again, there's one hint to us why this is so important, why they need a different, another alternative pathway to control the metabolic one. So the metabolic analysis showed in a high stage 3 and stage 4 colorectal cancers, they have a particular cluster of the lipid metabolic metabolites get enriched. And our single cell sequencing data also shows in a high grade colorectal cancer, GBE1 reaches the higher expression level. Not only this, along the HIF1 alpha keep accumulating, there's a switch to the higher expression of the GSK3 pathway and the fatty acid metabolic genes got upregulated. From here, there's still some validation experience still unworking, but we can see there's a hint, like can mimic 2 different reprogramming pathways mechanisms here. The first one is we already show you the canonical glycogen metabolic. When the hypoxia coming up, the GBE1 gets a higher overexpression by the HIF1 alpha trigger. Now, and the alternative pathways, the lipid metabolic reprogramming in a late stage of colorectal cancers, driven by the PPAR family, to driven the fatty acid pool to donate the energy storage and reach to the tumor progression. Here is our findings from the computational. We also not only using the computational part, but also we think beside the animal model, what's something this idea can donate to the clinical. So in the colorectal cancer, there's another very interesting molecular subtype called the CMS systems. So CMS system found 1, 2, 3, 4 different subtypes. The first one is for the microsata instability. So they have a higher chance to activate the immune response and the chronicle. But the CMS3 and 4 are the worst prognosis. This data from the Singapore data set also shows the worst prognosis of the CMS4. So we simply check the expression levels. We found in the CMS4, there's an enriched CMS4 expression levels of the GBE1, and along with the hypoxia and the glycogen signature. So in these signatures, the pathway also shows the same direction of the enrichments of the glycogen synthesis and the glycophospholipid catabolism and the Wnt signaling. So based on this, we're trying to find that is any reagent we can use to leverage these symptoms. So we decided machine learning models to simulated the different GBE1 protein structure through alpha-4-3 pipeline, and we have our proposed infant structure in the following project proposals. We use the expandable AI machine learning model, trying to distinguish any important features as an eigenvalue, which can predict the outcomes of the treatment. So we found several lists. We found the 27 compounds libraries. We identified 5 repurposing anti-diabetic reagents, which can bind to the GBE1. And these reagents, as examples, they have the specific binding with 14 hydrogen bonds, also the hydrophobic packing and the aromatic staking. So this binding is more specific instead of like non-specific just adhesion. Also, this is one of the dual targets molecule designs. So it not only can bind to the GBE1, but also bind to the PPAR gamma. So we use this as our lead to check with the functional assay. They have the really good cell death effect and directly inhibit the GBE1 enzyme activity. So using the expandable AI models, we can predict. We're trying to use this model to simulate and predict what kind of the features can be used to alterate these drug outcomes. The first one is not surprise the glycogen content and the GBE1 expression level, and also carbohydrate metabolic signatures and the lipid signatures. Surprisingly, we also see the particular CMS subtype as a contributor which can be an eigen parameter to predict the outcome of it. So make a quick summary. From the acute hypoxia to the prolonged hypoxia, energy got accumulated in a very early part. So when the prolonged hypoxia, the energy gets reduced, used. Once we use a dual target compound to inhibit GBE1 in a very early, they can stop this fate. Then, Then the second part, to inhibit the fatty acid pool, to further block the energy supplies and lead to the tumor eliminations. So here make a summary. Hypoxia is a core loop which triggered, not only triggered by the HIF1 alpha. We're trying to find the switch point to leverage in a particular molecular subtype of colorectal cancers. As a take-home message, we're trying to use the dual modulation to leverage the metabolic reprogramming to treat the colorectal cancer. And we're trying to expand and get more focus on the Asian population, a particular molecular subtype, to see if there's any more important factors we can find. So last and last, we very grateful to have the really good networking, really good collaboration under the MOHWC Cell Cancer Project type. And also we thank for the kind of data sharing from our international collaborators. Yeah, I will stop by here. Thank you very much.
Thanks for Dr. Xu's great talk. Is there any question?
Hi. Yes.
GWAS and TWAS.
Yeah.
So what's the GWAS hit doing? Is that
That's
is that
from
is that an EQTL that you found?
no,
there?
that's the TWAS.
Right.
The one is from the TWAS. Unfortunately, that in the natural genetic papers, authors didn't do the maybe that's the limited of the numbers. So we don't see the stratification or the adjustment from the populations. And then moreover, the data set is more like early part, like stage 2 or 3B. So like more early part. So that's why we can both see the weak significance in the carbohydrate metabolic, also see the lipid, but there's no like skewed or very strong signals was found.
So Fang-Chi, thank
Yes.
you for the talk.
Yeah.
I'd like you to clarify about
Sure.
the Asian connection.
Yes.
Okay. In terms of expression or in terms of metabolic modulation.
Yes. Yes. Oh yeah. From the paper data, they are like a half European ancestry, half Asian. But is that about our data set? Oh, sorry.
I thought you mentioned this GBE1
Yes.
has a connection with the Asian
Yes.
tumor.
Yes.
Yeah.
Yes.
I'd like you to clarify whether it's due to the specific expression or because we are eating different foods. Yeah.
Oh, yeah, that's the expressions. That's from the TCGA and the proteomics database. We can see that there's a higher expression level and a higher methylation levels in Asian population. Compared to the European ancestry, we reach the significant, but that's the expression level. But we don't know if that's because of the originate, like the race differences. Because in the large scale data set, we didn't see there's a good stratification, so there's no directly evidence to show it's from our ethnic or the race differences. So then in terms of that, they might be like modified by our diet as well.
So because of the limitation of time, and let's thank Dr. Chu's great talk again.
Thank you very much.